So yeah, let's get to it. I'm gonna try to tell you about what I'm doing on these projects that I mentioned and that you already introduced, Andre. So to start with the basics and what all of you probably already know and maybe even know better than me, through your lived experiences, we have OGT-CDG, OGT-XLID, OGT-ID, which are all the same. I'm gonna say OGT-CDG in this talk, and it stands for transferase congenital disorder of glycosylation. It is an X-linked intellectual disability syndrome. It's caused by mutations in the OGT gene on the X chromosome, and it is named like this because of the affected biological processes. Yeah, it's involved in what is called oglycogenation, which is basically a form of glycosylation where proteins are modified by adding a sugar. This process or this modification has a lot of different functions in the cell and can give rise to a lot of different symptoms, as we already know. But I'm gonna go back a little bit. It's a fairly new diagnosis, and it's a heterogeneous diagnosis. Like the association between mutations in OGT and intellectual disability has just been established recently or a few years ago. And here you can see the number of patients that were, like the accumulated number of patients that were discovered these different years. And in 2020, there were 13 patients that made the basis of the first characterization of the syndrome as a whole. There was this publication, and I'm going to show a table that is really busy, but don't get uneasy about it, because what I just want to tell you is that here we have the 13 patients, and we can see that there are a lot of different organ systems that are affected. It's a very heterogeneous syndrome, but also we can see that we have lots of missing data points. Whenever it says NA, it means not available. So basically, for some of the patients, we don't know a lot, or for some of the phenotypes that we see, or some of the symptoms, we don't have that information for a lot of the patients, some of the patients. However, what we can see is the main symptoms and intellectual disability, or IQ, that is down here. It can be seen in 13 out of the 13 patients, so 100% of the patients. Other strong phenotypes are developmental delay, language problems, and dysmorphic features in particular. I also want to emphasize and put it in a bit more visually nice way, and say again that we have a lot of different organ systems that are affected. It's both heart issues, facial features, digestive issues as well. But we still lack a lot of data at this point back in 2020 from some of the patients characterized. So it is still, at this point, largely uncharacterized. Since 2020, a lot more patients have been identified. In 2025, we have identified 21 new patients. And we are starting to see the phenotypes more, not strongly, but we're getting a better idea of what it looks like. And here we have, this is based on 63 patients, and the gray parts of the bars indicate that we don't have data from this number of patients, and the orange part is the patient not presenting with a given symptom or phenotype, and the blue parts are when a patient has a certain phenotype. And as you can see, we have ID as the most the most frequent symptom, and along with developmental delay and still dysmorphic features. So we're starting to get a little bit a better idea, but we still don't have complete data, so to say, which lead me to the reason why we want to do this natural history study, because we have bits and parts of information. We have this rapidly increasing number of patients that are now worldwide. It's a very heterogeneous patient group, so it's difficult at this point to say to what extent a symptom is just contained to a subgroup of patients, or if we just don't have the data to say that all the patients have the symptom, because the data is incomplete. Yeah. So, and please, actually, I wanted to say, please feel free to interrupt me at any point if something is unclear or you have any questions at all. Anyways, yeah, so this is the why we want to do the natural history study. We need a full characterization of this syndrome. And how we want to do that is through systematic and extensive data collection. That will, like, we have developed these questionnaires now that are based on everything that we know already. So these different lists of symptoms that we have already collected, we've taken everything together and developed 2 questionnaires, one for the clinician to fill out with their more technical skills and clinical evaluations, and one for the parents or the caregivers to fill out to give more, what can you say, the lived experience of what it is to live with OGT-CDG. And the status is that the questionnaires have been sent out to all of the clinicians that have agreed to participate in the natural history study, and we have received a few back, but I think this is a process where we need to figure out, like, because we really want it to be systematic and we want it to be complete, we need to go a bit back and forth to make sure that everything is filled out correctly and satisfactory. So what we hope and expect to get out of the natural history study is, as I said, to fully characterize this complex syndrome, but also to identify what are the core symptoms of OGT-CDG. We also want to identify what symptoms are shared between some or many of the patients, and what symptoms are unique to subtypes of patients maybe. And in the longer term, the aim or the hope is that this can help guide patient information and care by informing clinicians and patients and families, basically. Yes. Okay, so I'm gonna make a little bit of a shift now. So how can a change in a gene lead to a clinical symptom, or what we see as a phenotype? And this might be a little bit overwhelming, but I will walk you through it simply and slowly. So in all of our cells, we have the same DNA with the same genes that can be expressed or active in different cells. Genes, they can be made into what is called RNA, And RNA can be made into proteins that have a function in the cell and can have different functions in different cells. Proteins, they can be modified. And this modification can also affect their function within the cell. And all of these different functions that proteins have in cells are what make cells into specific tissues that make up specific body organs and have a function in the body and makes up the whole body and the whole person, right? Yeah, but I want to zoom in a little bit on protein modifications and explain this biology. I know that there's been given talks previously, so you probably have heard some of this before. I'm just gonna walk through it again. We have OGT and OGA that mediate this addition and removal of a sugar to proteins. They modify more than 9,000 proteins, and this modification or this process is essential for life, which is also why the balance between OGT and OGA is very tightly regulated. So what happens when a gene or the OGT gene is mutated? Well, it affects both the protein itself, but it also affects the proteins that OGT can modify with this sugar. And it can even affect this tightly regulated balance between OGT and OGA. And all of this, of course, can have an effect on the cell where the proteins either don't function as they should or function differently in any way. And because OGT affects so many different proteins, this can give rise to a lot of different effects in the cell and in the body and give rise to this very diverse range of symptoms that patients with OGT mutations experience. Yeah, and to go a bit deeper into the OGT mutations, again, if we have the OGT gene and it's mutated, actually, that's not what I want to say, but here I have a schematic representation of OGT protein. And as you can see, we have all of these are different patient mutations, and they are distributed throughout the protein, and they are also spanning across both the two main domains. One domain is called the catalytic domain, which is here, and the other one is called the TPR domain, which is here. And they have different functions. So one could speculate that a mutation in one domain of the protein could maybe give rise to one set of symptoms or downstream effects, and mutations in a different domain of the protein could give rise to different symptoms or different effects in the cell and the body. So these different symptoms, like one of the things that I also want to do with these studies that I'm working on is to link the clinical heterogeneity or the different symptoms that we see in the patients with biological signals or profiles or biomarkers, or what we want to call it. And these two can even also be linked with the position of the different mutations to, as I said before, to see if we can in any way subgroup the patients, or if there are any correlations between a certain biology that is influenced and that give rise to a certain set of symptoms. But anyways, to investigate this, we are doing this biomarker discovery study And the reason why is because although clinical descriptions are extremely valuable, they are a description of what we see, the end result, so to say. Whereas if we want to understand what is going on biologically, we can look at the biomarkers and in a way narrow down what it is that we're looking for. Because right now a lot of things are up in the air. So how we want to do this biomarker discovery is basically one in-person visit with the patient's treating clinician, where some specific tests can be performed, and blood sample collection and buccal swab. Buccal swab is on the inside of the cheek. And with these two relatively simple biological samples, we can analyze a lot of different layers of the biology and correlate this with the clinical data. So actually to get back to some of the things you said, Andre, is that I started this these two projects more than 1.5 years ago. I set out to try to get an ethical approval here in Denmark to enroll all of the collection sites across the world. But the rules have been changed and they're super strict, and it has delayed the project a lot. However, now we have the framework for the ethical approval in place. So we know exactly what to ask the clinicians to get them approved in our ethical application. And that is what we're currently doing. We're asking clinicians that have agreed to participate in the study to give us the documentation that we need to enroll them in our study. And if what you can do, please help nudge the clinicians. I'm doing what I can, but maybe if we attack from different angles, it will be even stronger. So, and also actually when we started out, the biomarker study and the natural history study was kind of one. Now we have divided them into two separate studies, because the biomarker discovery takes longer because of the ethical process. So what we hope to get out of the biomarker study is, one thing is to identify a reliable biomarker to assist diagnosis, because at this point, if a patient has a mutation in OGT and they don't know if it is the cause of the symptoms that they experience, it would be very valuable to have an easily accessible tool to see if it's this balance between OGT and OGA, or something related to the OGT biology that is disrupted, which would indicate that that the OGT mutation could be the cause of the symptoms. It can also, we hope to improve the biological understanding of what is going on. And also, as I mentioned before, to divide patients into subgroups, because this might help even look at severity or at prognosis in the longer term, obviously. And in the even longer term, we hope that it could provide tools to measure disease mechanisms or even responses to future treatments, but that's quite far down the road. So right now, the next steps are that these two projects, they have been initiated. I am following up as much as possible, or constantly, I wanted to say, to get the data for the natural history study, to get the clinicians and the patients enrolled for the biomarker study, and then obviously there will be practical steps like data analysis and at some point results. So, but with this, I just wanted to say thank you to everybody for making this possible. Especially, I want to thank the clinical collaborators that we have a lot of, and in particular the patients and the families, because your contributions are invaluable to this research. Yeah, and I'm happy to take any questions.
Thank you, Marie. This is super inspiring. My first question is, how far along are you for both projects? Do you have any, like, a progress bar or something? And I'm trying to understand how also that's a basically second question. How can we help? How can parents reach out to our clinicians? We need to basically provide the visibility that you've sent out this this request, and we just need from our end to support this and just follow up.
Yeah, well, as I said, like, both projects have been initiated, the emails have been sent out. I'm still like pinging clinicians that haven't responded if they want to participate, or like I know the clinicians are also very, very busy, so I have sent out emails to some that never got back to me. And so, like, I'm just trying to, because that's the other thing, we need a certain number of patients to be to participate in this, for it to be meaningful and for us to be able to say something that makes sense for both the research but also the patient community. So I think right now it's mostly, I don't know if there's a lot you can do, like, not sure, as I said, not sure clinicians, if you know any clinicians or can help each other. I have talked to some parents that are looking for a clinician where they can go and do, like, get enrolled in this study. And I'm not sure how to help someone in the US with this, honestly.
Sorry to interrupt. My son Kellen just had his neurology appointment. One thing I did talk to her about was, the fact that we are doing all of this research with you guys, and that, I discussed doing an MRI again, and she was good with that. What if there was, like, some sort of letter you guys could send to us parents that we could take to the clinicians that we're working with, and just, talk to them about which tests would be good to run for our kids to have more information available?
Yeah. I think that was kind of two questions in one, if I understood you correctly. So one thing is that if you parents can like give the questionnaires or go to the clinicians and say, hey, let's do this. And the other question is to find out what tests could be good to do for the patient group, right?
Yes, I mean, I know for Kellen, we already found out when he was little that he had delayed myelination. We were already following with MRIs, but once he's 5, of course, the myelination slows down. But I was able to convince her that it would still be good to keep a good eye on him with an MRI because he can regress.
Yeah. Yeah.
But if there's a test you guys think would be something we could do at our, when we go to these neurology appointments, then if we have a list of them, the parents will know what to ask when they go.
Yeah, and honestly, MRI is something that we discussed including in the study, but that we chose not to ask specifically for, only if data is already available, because we don't want to put too much, like, it's a big, if the patient needs to undergo anesthesia to have the MRI taken, it's not something that we want to put on you guys, right?
Mm-hmm.
But obviously any data is extremely yeah.
Okay. They were telling me in the beginning to follow Kellen's A1C. Is that something that's recommended for OGT-CDG kids?
Come again, sorry?
To follow their A1C, because apparently that can be affected.
I'm not sure what that is. A1C?
It's normally used for diabetes.
Okay, yeah, I am not familiar with that,
Okay.
honestly. So I cannot.
They
Yeah.
were their doctors that are not CDG doctors. So, I mean, it might be something that was in the literature for different CDGs that, and maybe it doesn't affect OGT.
Yeah.
Miranda,
Okay.
I think that, and maybe if other are like, oh, GlucNet researcher are on the call, but I don't think I've ever seen anything on watching A1C for, or changing A1C glycosylation level based on a GlucNet modification.
Okay. Well, that's good to know.
But I mean, all this, it's not like it doesn't hurt to follow your A1C anyway.
Yeah.
Right? Like it's part of the blood draw, like they do, and it's like one more test. I don't think it hurts, but it's also unlikely that too young like this, if it doesn't have any diabetic history, that will affect anything.
Is there any blood work our kids should have done? Sorry.
Oh, Dan is on the call. Maybe you can answer.
Yeah, well, I'm a bit dark. I'm like more like a phantom in the dark here. I'm sorry about that. It's dark here in Denmark. I just wanted to add one thing, and that is that, you know, unfortunately, this CDG in the abbreviation OGT-CDG is really unfortunate, because people start reading about other CDGs and this isn't. So maybe in other CDGs it's important to follow whatever parameter. This has absolutely nothing to do with that. So, you know, we can't use the standard assays, for instance, the ferritin electrophoresis mobility assay that people use to diagnose CDGs, absolutely pointless for OGT. And so there's still a lot of parents that start reading about CDG, and they think that OGT is just a subclass of a CDG, and it's not. It's completely different. I don't know whether you know, Stephanie wants to comment on this as well, but I think that it can lead to confusion and wasted energy, basically.
Okay. Well, that's good to know.
Talking about this, Dan, like on the page that we're doing for the database, we call this OGTX lid, which I think is part of, like, you guys is also thought that, you know, we should not call this CDG because then it leads to more confusion, that it's a classic, it may be more close to a classic CDG than anything else.
Yeah, no, I agree. Yeah.
Marie, do you have a timeline for either of these projects?
Not a fixed one, but I have, like, I think I mentioned that the NHS, the natural history study, has a bit shorter timeline because we can actually start collecting the data right now, right? Whereas the biomarker study, we need to get these ethical approvals in place before we can do anything. And I think it's very frustrating because I know a lot of you guys have, like, we keep getting asked, like, why can't we just send a blood sample? And we need to do this the right way. And unfortunately in Denmark, that means it takes a lot of time. But as I said, we have the approval for one patient, so we know exactly how to set this up, and we know exactly what to ask the clinicians for, and we have asked them now. We just need them to get back to us, basically, with this information so we can get things going. But I would say for the natural history study, I would love to have some preliminary results ready in, let's say, 3 months or something, right? But it depends how fast do the clinicians get back to us, because if I have 10 responses in 3 months, that's not enough, and it's not meaningful to go out and present that, I think.
Yeah. So I think that you could absolutely use this group of people, right? So you can use us because we have connections to our parents, and we can ask them to go out, as Miranda said, and ask them to fill out any forms. If you need to put us on CC, or if you want to send us, hey, these are the clinicians that I've sent these emails to, we can start following up with them, or whatever you feel the best way of doing this, because I also understand that could be like, you know, a researcher to a clinician communication, and patient could just like worsen this communication. So whatever you feel reasonable, use us as an opportunity here. Hey, Catherine. Catherine has a hand up.
Yes, I wanted to comment two things. It was first regarding the evaluation. Yes, I'm agree with Dan that I have evaluated also our patients regarding like a typical CDG biochemical guidelines, and everything has been normal. It has been only this one issue with a low level of activated T lymphocytes. That's it. So there are not much what to look at. But as a basic, we still have to evaluate if a patient is coming regularly, just a basic biochemistry. But regarding your invitation, it's a bit, because you have sent it only once, I just looked at the date, it's on my to-do list. It was a bit bad timing before Christmas. And I was away almost 3 weeks, and I'm still trying to keep me on track. So I really suggest now to repeat it to clinicians. It's just a suggestion, yeah. And thanks for this description. I already sent it to my patients' parents forward to fill parental questionnaire. This is also another option that you can share those, because it's a 2 separate questionnaires. One is for parents, another is for clinicians. But the parents can fill out their side, and then they can push their clinicians to fill the second part.
Yeah.
If they are agree, if they know already that this is available.
Yeah, I think that's a good idea, Catherine, to like now I've sent it out, and actually something that's on my to-do list is to send a reminder to the clinician, because it was just before Christmas, basically in the middle of December or something like that. So that's something that I wanted to do, and I'm gonna keep doing that. I don't know, once a month or something, until hopefully I get a lot of responses and a lot of beautifully completed questionnaires. But I think it's a good idea to also, I'm not sure about the ethical aspect here. Dan, maybe you can pitch in a little bit, because I don't know, like obviously I'm part of this community, but ethically to send data to patients directly, I don't know if that's, and maybe Catherine, you know about this, because
you can send only patients questionnaire. You cannot send them a professional questionnaire, but patients are legally, they are aware to share any data of their kids, or patients can share their data, and some of registries, international registries, are based in this way, that patients are sharing their data into different registries. Because if we are recruiting, for example, patients to any kind of studies, we are always asking the consent if we are sharing clinical data, because those are confidential.
Yes.
So it may be not ideal if you are in a situation that, for example, you have 20 answers from a parent, but those clinicians have not responded. But maybe on a third step, you can send directly, "Oh, Catherine, your patients already filled out this questionnaire. Please fill out your side too." It would be very nice to see comparison. But ethically, you can distribute it directly, because they are answering only their questions.
Yeah.
And I am not correcting their questions. Answers. I can only be responsible for my answers as a medical doctor.
Yeah, but that's exactly how we want it as well. So that's yeah. And I think, like in Denmark, at least, I know that the rules are that I can perform this type of questionnaire without getting consent from the patients. But
yes,
for the
correctly.
biological sample collection, I need it.
I looked it through what you sent, 3 documents, and on a third one, I have to confirm that my part is authentic and objective. But patients can, by rules, can fill out the questionnaire freely. They don't have to sign the consent. But as a whole, I know you have it already. As a whole, you need a research proposal and confirmation from your Danish ethics committee that you are doing such kind of a study. Yeah. And I know that you have it.
Yeah.
Marie,
Yeah.
thank you, Katrin. Marie, I see that Kenia has asked a question in chat. Kenia recently joined the community. She's from Poland, and she's asking, do you see any connection between OGT and MECP2? And do you know any other cases of like similar to that?
Well, I think that MECP2 is an intellectual disability gene in itself. So this one specifically, it can be difficult to know exactly which gene is contributing more, or if it's the combination of the 2 genes. We haven't performed any studies specifically for these 2 variants. So we don't have any anything we can tell about this. Yeah.
If I can also maybe add something to that, I found the light.
Yes.
Andre, is that actually it's one of the things that Marie didn't mention, is that the more patients that we have, the more observations we can make of mutations in other genes that occur with OGT-CDG and maybe make the symptoms worse. This is interesting for a number of reasons. It actually hints at the potential mechanism, but also can explain why some patients have much more severe symptoms than others. But there too, like Marie said, this is only going to work if we have a lot of patients. And I know that I go around in my talk saying, oh, you know, we have 90 variants, but if in the end only 15 clinicians respond, we cannot do anything. We are actually, to go back to Kin's question, we are actually already starting to notice a little bit a co-occurrence of some mutations in some genes that are a bit similar to this MECP2 with OGT-CDG, and we are actively investigating that. It's super exciting because basically that, again, as I said, it can give us a focus on what processes to investigate. So the more patients we have, including patients where, like Kin, where we have OGT-CDG mutations, but also another mutation, in particular, if this seems to make the symptoms of the patients worse, for us, that's really interesting.
Makes sense. I'm trying to translate this to Polish because Kenia is not very well with English. But Dan, thanks for that. All right, we're almost at time. Do you have any last questions to Marie or Dan or Katrin while we have them here? Marie, I have a question about the biomarker. So this will require also a significant lift from all of us to deliver the patient samples. Has this already been started? Is it also part of this framework that you've shared, that you've tested it on one patient and then you'll just have to roll it out on everyone else? Is it related to that?
It's only the ethical framework.
Yeah.
So we have gotten the patient sample from this patient, and we are storing it and waiting to get more samples, because it's also, if we process the samples in different batches, it can introduce technical variability between the samples, and already we are working with a very heterogeneous disorder. but also a very heterogeneous patient population that are different sexes, different ages, different ethnicities, different genetic backgrounds, different upbringing, environmental influences. So we expect to see a lot of variability in the data and in the blood. Yeah, so I don't know if that answered your question, but we haven't performed any test on patient blood samples yet. We have performed some what is called RNA sequencing, bulk RNA sequencing on whole blood from mice, from one of the mouse models of the disease that is in Dan's lab, to also test the tubes that we will need for transportation of the blood from across the world to Denmark.
I see. Okay. Yeah, well, keep us posted on how that progresses. And
Yeah,
again,
sure.
if anything is needed, just let us know. For example, we went to New York to Dr. Eva Marava, and we provided our blood samples as well. So I think it is already stored there, and I'm pretty sure that she stores more of such blood samples for other patients. So it's already it's a matter of figuring out when and how to transport them, like in the ethical committee. So whenever it's ready to be done, I'm sure that she will help collaborate.
Yeah, and on that note, I want to ask: did you also get a blood sample from like one of you?
Siblings. Yes, sibling. Yeah.
Perfect.
Because we kind of saw that this was coming, and we provided the sibling sample.
Yeah.
Yeah.
That's great.
And on another note, will you work on patient-derived iPSC cells, or you don't need to do that at this point? Well,
go
ahead.
That is not the plan at this point. It doesn't mean that it's not going to happen, but with the figure I showed you, this schematic representation of the OGT protein, you can also see that almost every patient has a unique variant, right? So we obviously can choose one and work on that, but maybe it makes sense down the line. But we're gonna start with these different layers of biological. Biological layers, and see where that takes us, because I think, Dan, what you also implied with these co-occurring variants is that right now all doors are basically open, and we want to try to figure out which one are the most likely that we find something if we go in there, and this is a way to do that. So the short answer is no, that's not the plan. The long answer is maybe down the line.
Yeah. Makes sense.
Okay, so I did mention this to a couple people already, but we were working on, so for those of you who don't know me or us at the lab, we have, we're the host and curator of the OGT-CDG database, which is this resource for mostly the research community, try to find OGT-CDG protein and try to improve research in OGT-CDG. One of the things we've put up and try to work on, thanks to Dan, Lance, and Mary has been instrumental in sending us some data for this. We've started working on all the variants described for OGT, and this bit here is really the research side, but we wanted to do something for patient as well. So we've created that OGT-CDG page. And so now this is open, you guys can see it. If we have a way to put this on the Facebook page or something, Andrea, you can do that. But basically what we're trying to do is a page so patient can land on this and parents of patient can land on this, and then have quick information about what OGT-CDG is, but also have a state of all the variants described for OGT-CDG so far, which is where we're bridging a little bit the research side, try to get the data from Dan's lab through Mary and then through Lance's lab as well. try to pull everything on one place. So here now, what you can see is we have all the variants represented. So every single variant that have been described that we know of are represented on this, and we have some quick information about where and what the OGT is and what the different domains are. Try to make it an easy way for people to understand. We do link the QOGT as well. So there's also a direct click. If new patients are trying to find information, they could go to that website and then link to you guys on the patient side. And then there's more after this that explain what a GLICNAC is. One of the things that we did is we have a translation aspect to it where people can choose their language. And so hopefully that's going to break a little bit the barrier of like people if they don't speak English, we have access to everything, all the different languages. So yes, I just wanted to share this. So again, and I'm guessing also for maybe genetic counseling, if they end up like trying to find information about OGT, they can get there and put you guys in link with the new incoming patient. I newly identify OGT-XLID. So hopefully that helps.
This is great, Stephanie. A quick question. Well, do we have like severity of phenotypes attached to those variants?
So, okay, no, we don't have any clinical data attached there. On the research side, what we have for each variant, and there's more than just the OGT-XLID variant on this, but the only thing we get out of kind of like the research aspect is we have severity of mutation, which doesn't always correlate with severity of clinical presentation, but you'll get if this is a damaging mutation, like is it going to change the protein in a really dramatic way? And those kind of information are represented here, but it's not. It doesn't often correlate necessarily with like the severity of the patient like symptoms.
Gotcha. Right.
But yeah, we don't have those information, and the only place I feel like this may be put somewhere in databases that we could have got is probably ClinVar, but ClinVar is really not up to date as far as the OGT-XLID variant. So
I see.
Okay.